Yeah, so the last part of this chapter is just to go about what are the animal or living models that scientists have been using to understand how we work. And sometimes if you see some researchers working on fish or working on bacteria or working on yeast, you may not understand why is it important to understand how bacteria work. What's important is to know how we work. But actually, if you think about it, at the origin of life, there was maybe only one type of cell, and this cell evolved to give many different types of cells that we know today. So knowing how, for example, bacteria work helps us understand how we work as well. Especially Escherichia coli, this E. coli dijoking, it's extremely important because everything we know about molecular biology was at some point studied in E. coli. How genes are transcribed into RNA, how RNA is translated into protein. All these things were originally studied in E. coli. So this was very important and is still very important in molecular biology. Maybe every day in the laboratory we use E. coli. But of course, sometimes you want to understand how eukaryotic cells work, because there are things that bacteria do not have. As you know, bacteria do not have mitochondria, they do not have organelles, they do not have nuclei. So you want to understand how these things work, and you must actually change from E. coli. And this is where yeast, Saccharomyces cerevisiae, this is the yeast that is used for bread or beer, has been one of the most successful models to study the eukaryotic cell. And that's just it. That's just yeast. It's one single cell and it can divide, giving many cells. And in one cell, it has everything that our cell have. You know, as I said, nucleus, mitochondria, just everything. Simply, they grow very fast. They are single cell organisms, so it is very easy to study these cells. And one of the most famous discoveries using these Saccharomyces cerevisiae, this is this one, right? S. cerevisiae. A scientist called Paul Nurse, maybe you have heard about Paul Nurse. He studied many years ago how, cell division, cyberbulletsu, what controls cell division? What will make the cell divide? Okay, so he tried to understand how these things were regulated. And because it's a single cell, it is very easy to make mutants. You can destroy specific gene and see what is the function of this gene. Or you have many cells in front of you, and some cells may be a little bit sick. Some cells may not be able to divide. So then you can study what is strange in these cells and which genes are broken. So this is what he did, and he found some cells. It's a different type of yeast. It is still yeast, but it's called Schizosaccharomyces pombe, S. pombe cells. And these cells, they are alive, they can divide, but they are a little bit strange. Is that if you raise the temperature, they cannot divide. If it's cold, they can divide. But if it's hot, they cannot divide. One gene is a little bit strange. And they discovered, by studying these sick cells, they discovered that one gene
CDC2 was not working. It was a mutation in a gene called CDC2. And this CDC2, later on, it was understood that it's a gene that regulates cell cycle, okay, cytokinesis. But then, in the beginning, they didn't know. So, they had these cells, and they were in a tube, and they were unable to divide at one temperature. But what they did is they took another yeast, and they extracted the DNA of this another yeast. And they cut the DNA into small pieces, and then they mix these cells together with the DNA from a different type of yeast, different species. And then they treated this a little bit. And after that, after incubating the cells with DNA, then they put it into another dish at one temperature. And something happened. Most of the cell could not divide. Of course, because they could not divide in the beginning. So most of the cell will not be able to divide after incubation, but still a few cells could divide. Somehow, these cells changed by something. They could recover the ability to divide. And what they did is then they looked at what is the DNA of the cell that can divide. And in these cells they discovered that the CDC2 gene of S. cerevisiae was inside these S. pombe that could divide. Do you understand this one? So the S. pombe cells became transformed by the DNA of S. cerevisiae. So the DNA entered into the cell and they could recover the ability to divide. Wakaru, sorry.
Yeah.
Did you understand this one?
Yeah, probably.
Yeah, probably, yes, probably. But, yeah, this is okay. You kind of understand, Mama, this is yeast, and this is yeast. It's the same kind of animal. It's just like a cat, and maybe another species of cat. Or between a cow and a horse. まあそんな違うではないね. It's a little bit similar. But then they did the same experiment, so exactly the same experiment. なんか来ました。大丈夫ですか? They did exactly the same experiment. So they had these yeast, these S. pombe, who could not divide. They were mutants. And what they did, instead of mixing these cells with DNA from a different yeast, they mixed these cells with DNA from human. Completely different. Yeast and human, it's two completely different. And then they mixed these cells with the human DNA, and after incubation, they transferred the cell to a warm medium, and they could see that some cells could divide. They recovered the ability to divide. So what do you think happened? What do you think happened? Keep in mind that in this case, this is not yeast A and yeast B. This is yeast and this is human DNA. Completely different, right? Completely different. So what do you think happened? Why some cells, after being mixed with human DNA, why some cells could be repaired? Why do you think?
Humans have CDC2 gene.
Yes, this is exactly what happened. So they analyzed the cells that could divide at one temperature after, and they discovered that all the cells that could divide, somehow they had incorporated a copy of the human CDC2 genes. And if you check the sequence between human CDC2 and yeast CDC2 gene, you can see this one. This is not DNA, because you have F, G, L, A, R. What do you think this is?
Peptide.
This is a peptide. This is amino sequence. And you have the human local sequence of the CDC2 gene in human, and the CDC2 gene of Schizosaccharomyces pombe and Saccharomyces cerevisiae. And you can see it is very similar. F, G, L, A, R 全く同じ。SA はちょっと違うけど、まあ、Saccharomyces cerevisiae and human is the same as well. FG, this is also the same. So it's very similar. So this is one of the evidence that actually we have the same origin. Yeast and human have the same origin. We share the same sequence of our protein. So one question now is why the sequence is not 100%. So what does this experience mean? This experience of having a CDC2 gene from human can repair mutant yeast. What does it mean? What does this experiment mean? Can you tell me? Can you explain what it means? Can you explain what it means? It's not easy. It is not easy. What is the meaning of the previous experiment?
Not 100% same is not important.
Yes, this not. You don't need to have 100% of identity to work. This is true. What else? Without thinking of the sequence, what is the meaning of this experiment? Think, this experiment means that you put human DNA into the yeast and it's working. So what does it mean? What do you think it means? You put yeast, you mix with human DNA, and some yeast, they are repaired by human DNA. But what does it mean? They are repaired by human DNA. If you think about it, it's very incredible that it's working. It's very incredible. What is the biological meaning of that? What do you think?
Proteins have not so important region.
Yes, that's for the sequence. This is for the sequence. So we will come that later. This is for the sequence. But without thinking of the sequence, what is the meaning of this experiment? Yes, somehow you know these cells recovered because they incorporated some human DNA that contained the information for the CDC2 gene. So, okay, this is fine, but what does it mean? It's a little bit like, for example, I go to China, for example, and a Chinese person gives me a book written in Chinese, and I open the book and I cannot understand anything. That's a little bit similar. That's the opposite of this. I am given information, but I cannot read information. It's completely different from what I am used to. No, I can understand French, I can understand English, I cannot understand Chinese. But in this case, what's happening? In this case, what's happening? And if you think about it, it's really incredible because yeast and human, there is maybe billions of years together. You know, they separated very early during evolution. What does it mean? Think.
Maybe you mean codon is the same.
More generally, you are talking about codon. This is already a kind of it's a difficult concept that some people don't know. More simple, what does it mean? It's the same idea.
Genetic code is same among species.
Yes. The human genetic code and the yeast genetic code is the same. It's the same. And you think, well, everybody knows we are all made of DNA. Okay. But if you think about this, yeast can understand human DNA. That's incredible, isn't it? You put human DNA, you insert into the yeast, and the yeast cell will understand the human DNA. That's really incredible, I think. And your cells can understand DNA from any organism. You have cells, and you put mouse gene, your cells will understand the mouse DNA. You can put into your cell Drosophila DNA, fly DNA, and your cells will understand Drosophila DNA for the most part. This is really incredible. Okay, this is really incredible. DNA from different species is understood by many different cells. So this is really, really important. And then now we come to the sequence, and you can see that, yes, obviously, the yeast cell can understand the human DNA because actually the sequence is not so different. We are all made A, G, T, C, right? We are all made of the same DNA. But as you said, you notice that some regions are conserved and some regions are not so much conserved. So what do you think it means? Why these regions are conserved and why these regions are not conserved? What is the meaning of that? Why do you think some region of the protein are very strongly conserved? Why others are not conserved?
The green region is very important, but the white region is not important.
Exactly. What do you mean important? What do you mean important?
The white region is unique to each animal.
Yes. Yes, this is not so conserved, right? So it's different between different animals like yeast and human. Go. So it means it's not so important, right? But what do you mean by not so important? What do you mean by important? In terms of the protein, this is the protein. This is not the whole protein. It's only one small part of the protein. なんかね、まだプロテインがあるね、ここも。 So why do you think this is important? Why do you think this is not important? What does it mean being important or not for a protein? Of course, maybe I'm hoping you already know how protein and enzyme work.
Being important means using for bonding.
Yes, for example, maybe this region is very important for the CDC2 to interact with its partners in the cell, or to interact with DNA, or to interact with other proteins. What else? Not only for interaction, but what else? Think about many proteins that we have. What might be important, especially for enzyme, if it's an enzyme? For reaction, not only to interact with other partners, but also for the chemical reaction with partners. For example, if this is an enzyme that catalyzes a specific reaction, like for example a kinase or something, it might be important for the chemical reaction, okay, for the activity of the protein. While the white region may not be so important because they are only kind of some not really structured region and not important for the shape of the protein. Okay. While this might be very important for the activity and for the shape of the protein. Okay. If you think about this, I show you this figure from a previous slide. It's great to study bacteria, but this separation between bacteria, plant animals, and different, it's about 3 million years ago, so it's a long time in the past. Okay? So sometimes you want to look at something that is a little bit closer to us. And the separation between all these plants, animal, and fungi is 1.5 billion years ago. Okay, this is quite long. So sometimes you want to study something that is a little bit closer to us. And for example, the evolution of different plants is only 200 million years ago. So this is much younger. Okay, this is much newer kind of invention. So sometimes it's great to study single cell. This is great, but sometimes it's also nice to study an organism that is closer to us in terms of being made of many different cells, like a plant. Okay. So for the plant, this became very famous. This is Arabidopsis thaliana. It's written here. Arabidopsis. And many scientists in Kyoto, in the world, many scientists are studying this plant because it can reproduce very quickly and it's very easy to study. But plant is great. We can understand many things about ourselves by studying plants. What else can we learn from plants? Why is it also important to study plants? Not only because we can understand maybe how it works, how we work, but what other reasons are there to study plants? Why would you study plants? Why would scientists decide to study Arabidopsis? Why it is important? Why it is important? Why it's important to know about how plants work and how they grow and how they produce tomato. Why it's important? We don't produce seeds. We don't produce, well, we do, kind of, but we are not plants. So why it's important to study plants? What do you think? Why would you want to study plants? It's almost as if asking why there is a nogakubu. It's almost the same thing, right? It's because we need plants to eat, and it's useful to understand how plants work, because then we can maybe make plants that can resist in the desert, for example. We can make plants that can produce many, many seeds, so we can produce a lot of carrot, or we can make rice that is very resistant. So all these reasons are also valid to study plants. Okay? Not only because we understand how we work, but also because it's interesting to know how plants work for commercial and for the society, okay, for the benefit to society. But okay, so that's about plants, but let's say you want to study animals. You want to know how animals work. And modern animals have been very diverse. Scientists have studied mainly a few of animals. Flies, Jojobae, Drosophila, fish, worms, and mice. Mainly, these have been the most successful models for biology. Let's see why. Drosophila. Drosophila has been a very successful tool for genetics. Almost all the genes of Drosophila have been studied by mutation. So you mutate one given gene and you see what happens to the fly. What is the function of that gene? You just remove it and you see what happens to the fly. Why is this easy to do with a fly? It's the same reason as why it is easy to study bacteria. It's exactly the same reason.
They very quickly reproduce.
Very quickly they reproduce. Extremely quickly they reproduce. You have millions of flies within a few months. So it is very, very easy to reproduce. And it's also small, very small. You can have millions of flies in a small box. But if you want to have millions of mice, you need a huge university. Okay? So this is just the advantage of the fly. So most of the things we know about how genes work, they were, in eukaryotes, they were probably first studied in flies. For example, I know circadian biology, the Tainaidoke. First, it was understood in flies, how the circadian clock works. It was discovered in flies. Okay? So this is only one example. And also, maybe this is something we will see later, but if you think about why the fly has wings there, why the fly has 1, 2, 3 pairs of legs, why the fly has one eye here, how is this controlled? Why the wings are not here? If you think about it, this is incredible. The fly starts as one cell, like us, one cell. But at the end, the fly always has here one pair of wings. And here, although you may not know, but this is also wing. All insects have 2 pairs of wings. But in flies, this pair is only developed as wing. This is only for balance. Okay? But it's also technically a pair of wings. So why? How is this regulated? And this is understood. Scientists have studied how the body of the fly is built by the gene. And they have shown that if you take a gene and you put it in a different location in the genome of the fly, then suddenly you have an eye here. And they can make flies who have maybe 4 wings. These kind of things. They have been understood in flies much more earlier than in mice or even in humans. C. elegans. That's another type of animal. This is a worm. Okay, this is a worm. And when you look at this guy, it may not be obvious why it is important. It's just a worm. By the way, this worm, you cultivate it on a small dish that contains content vitamine. And then you give to this. What does it eat? Do you know what does it eat? 何を食べるこれ？あの虫は何を食べる？ Sand, no. Maybe in the wild, if in the ground, it can eat sand, but it doesn't really eat sand. It eats something that is in the sand. Bacteria. In the laboratory, you give this mushi, you give E. coli. It can happily eat bacteria. But the importance of this worm is that it is always made of 559 body cells. Scientists know, they have discovered that if you take 100 worms, adult worms, they are always made of 959 cells. This is very precise. How many cells do you have? We don't know. We can only kind of calculate. But we know 959. So these worms are very important to understand how for example, there was not so long ago, there was a Nobel Prize awarded for autophagy. So cells, sometimes they decide to die because they have suffered a lot of damage, and the cell can decide, okay, I'm going to kill myself to allow the animal to live, because if I survive, it's not good for the animal. So these worms are a very useful tool to understand how the development, the life and death of cells is controlled. Okay? This is very, very useful because it's always made of the same number of cells. So, for example, if you make a mutant of this worm, and then you notice that it has much less cells, then you know that the genes that you are studying is something that is very important, maybe for how the cells survive and things like this. So this is very useful work. Fish. Do you know this one? Do you know what is the name of this fish in English? It's a very famous fish. 食べられないけど. It's a zebrafish. This is the zebrafish, and it's very useful, not because it's a fish, but it's because during the first 2-3 weeks of life, it is completely transparent. You can see through. You can see inside the brain. You can see inside the body. And because it's transparent, it is very useful to study how the body is made and how the body develops during embryonic development. Because how the fish develops is very similar to how we develop from egg. Okay? But if you study mice, the embryo is not transparent, so it's more difficult to study. Here, it's completely transparent, so it's great. And now, this is a comment also on the biological clock. Some of you may know that of course, in our body we have a clock, right? And the clock can keep time inside your body, but light is very important. Your clock reacts to the light, so that the time of your clock is the same as the time of the day. Okay? When it's the day for your body, it's the day outside. And this happens mainly because of the eye. Your skin cannot see light. your skin does not react to light. Okay, in terms of the clock. Your clock reacts to light only because your eyes send information to the brain about, hey, now it is the day, now it is darkness. But this fish, it's transparent, completely transparent. And it was discovered that every single cell can directly respond to light. Each cell is synchronized to the light-dark cycle because it's transparent. It doesn't need, like us, to have eyes like this. Of course it has eyes, but because it's completely transparent, then it's very easy for each cell to be able to receive light. Okay? Your brain cannot receive light because it's not transparent. Okay. But of course, you can study mechanisms in plants, you can study mechanisms in fish, in worms, in flies, but sometimes you want to study human. Right? You want to study how we work. But studying humans is not so easy because you cannot have human in a cage in your laboratory. This is not good. Right? And it's very expensive. But it's not good not because it's expensive, it's not good because it's not good. Okay? And you can say, okay, but you can study monkeys. Yeah, okay, you can study monkeys, but it's also not like a kawaiiね. And monkeys ものすごく高い。1匹は100万円1匹。すごい高い。So you cannot make a lot of studies, but you can use cells. You can use cells from human. And in the laboratory, in our laboratory upstairs, we have every day we are using human cells. And they were extracted from maybe a cancer patient 30 years ago or something. But still now we are using these cells. And they are happy. They are dividing and they still develop in a dish. But it's still human. And we can investigate what is the effect of drugs, how genes are regulated using these cells. And it is very useful to actually understand how we work just by studying human cells. Alright. So here you have different types of cells. You have fibroblasts. This is like almost cells that are important for the structure of your body. Okay? You have myoblasts. These are cells that have a relationship to the muscle, kiniku no saigo. And then we have epithelial cells. But this is different type of cells. Epithelial cells, this is like the cells of your skin. Okay? And you can see they are a little bit different. The shape is very different. And look at this one here. You can see the cell is here, right? That's the cell. And you can see some sort of weight. This is very special. This cell is cultivated on a trampoline mitai. It's a kind of plastic film that can move. Okay. So this is a film. Please look at the cell. And it doesn't work. But it's going to work soon. ちょっと待ってね。I don't know why they did not work. Okay, that's the guy. Sorry, maybe somehow the film was too old. Here, the movies I showed you before, it was cells moving. But in this case, the cell is not walking. The cell just moves on the same spot. And you can see it's very regular. What do you think this is? What do you think this cell is? Just looking at how the cell moves and how regular the cell is. What do you think this is?
Heart muscle.
Yes, it's a cell that has been extracted from the heart and just cultivated. And you can see, even though it's not in the heart, still the cell keeps on beating. And we can study many things with cells like that. For example, some medicines that may treat heart attack and things like this. These cells may be very useful. Okay, and now just to show you that although we are different, we are essentially the same, as I told you many times during the first part of this lecture. And one of the evidence is that for many genes, especially in mice and human, if you mutate one gene in mice and you find some mutation in humans of the same gene, very often the effect on the animal will be the same. For example, there is a gene called KIT, which is a gene that is involved in skin development and pigment. Okay, and then you can see this little girl here. There are some mutations in the human population. Some patients, some humans have a mutation in this gene KIT, and they are really characterized by white spots like this, always at the same place. And then, if you actually mutate the same gene in mouse, you get exactly the same. You get exactly the same. And for many genes, it's like that. So sometimes you have a mutation in human, but you cannot really understand what this mutation does, why these humans are sick. Then you can do the same mutation in mice, and you will see that actually the mice suffer from exactly the same disease. Koketsu-atsu toka, gan toka, onaji. So then you can use mice to actually study all these diseases and find treatment for these diseases. And that's another evidence that we actually share the same origin and we have essentially the same, really the same genes. Okay. And this is one of the conclusions that comparing genome sequences reveals life's common heritage. So this is again the same thing, but it's very interesting to compare different living organisms. So here what you have is the nucleotide pairs per haploid genome. Do you know what means haploid genome? How many pairs of chromosomes we have? Humans. How many pairs of chromosomes we have?
23.
23. How many chromosomes we have? It's different, right? We have 23 pairs of chromosomes. How many chromosomes we have? We have at least 46, right? Because we have 23 pairs. So what means haploid genome is that you take only one copy of each chromosome. So you only check what is the total size of one genome for each chromosome. So you don't take into account how many copies of each chromosome. You take one copy of each and you measure the length of the total DNA. This is what you have here. So the amount of nucleotide pair per genome, okay, for single copies of chromosome. And then you look at different animals. Human, among mammals, bird and reptile, you can see that's about 10 to the power of 9 to 10 to the power of 10 in terms of the amount of nucleotides that we have. Human is about here. Okay. You can see that amphibians, fishes, they are, the variation is much broader. And this is the frog, this is the zebrafish. Very strangely, you see that some of the fish and amphibians have a longer genome than human. Some of these amphibians or fishes, they have a very, very long genome, 10 to the power of 11 nucleotides. It's huge. Look at maybe protozoans, these single cells that I mentioned earlier in this lecture. Amoeba, very big, even bigger than amphibians and fish, but it's one single cell. How can the DNA of that single cell be bigger than our own genome? Bacteria and archaea in general, they have much smaller genome. Okay? But then you can also check something. So what you have here, this is the genome size. The genome size. This is a little bit similar to the previous figure, but maybe it's easier to understand. So human, 3,200 times 10 to the power of 6. Okay, that's the genome size in terms of nucleotide pair. And then you check E. coli. Now, it has about 4 or 5 million pairs of nucleotides. So there is a big difference between this. But then if you check the approximate number of genes, human has about 30,000 genes. A bacteria has about 4,000, 4,500 genes. So the difference between this and this is very big. But the difference between this and this is not so big. And we can compare Mus musculus, genome size is different, but about the same number of genes. Drosophila, the fruit fly, very big difference here, but that's only twice the difference. Okay, you can see 200 times 10 to the power of 6, 2,800 times 10 to the power of 6. That's about more than 10 times difference. That's about 2 times difference. So the amount of gene and the size of the DNA, it's not always linked. So what do you think happens? You cannot really make a linear relationship between the size of the genome in terms of how many nucleotides there is and the amount of gene that we have. Why do you think this is so? How can you criticize this information? Can you criticize this?
Non-coding region, not creating protein region.
Yes.
Is bigger in human.
Yes. Yes. So this is usually what people say. If maybe the genome size in human is much bigger than that of E. coli, but the amount of genes is not so different. So maybe the amount of junk DNA is bigger in human. And the amount of genes that code for proteins almost is the DNA of the Escherichia coli. It doesn't have junk DNA. It has only protein coding genes, while we have a lot of elonite DNA. This is what people say. This is what scientists maybe 10, 20 years ago were saying. That junk DNA in human is very big. We don't need this DNA. Maybe because it's old or whatever. Is it true? Do you think it's true? Do we have a lot of junk DNA? Do you think it's always true? Because that means that these protozoans may have a huge amount of germ DNA. So, なんかおかしいね。なんかおかしい。Do you think that there is a lot of this DNA that we actually do not need?
Junk DNA, so-called junk DNA region,
So-called junk DNA
is important for regulate the expression of DNA.
Exactly. So junk DNA means わからない DNA. We don't know what this DNA do, but it might have a function that is regulation, or maybe what is a gene? Can you understand really what is a gene? We know a gene codes for proteins, but we know now that there are many RNA that do not code for protein. There are RNA that regulate the expression of other RNA. microRNA とか long non-coding RNA とかいろんな種類がありますね、the RNA は。So it's not really junk DNA. I think probably the genome in human is very big because there is a lot of regulation on its DNA. Okay? So that's very limiting to say that there is a lot of junk DNA. There is a lot of DNA we don't understand the function of, but saying it's junk DNA may not be essentially correct. That being said, for a very simple organism like E. coli, they are very simple. They are extremely simple organisms, just one single cell, no organelles, no nucleus, no whatever. So these E. coli, they are extremely efficient organisms. They are very efficient. Okay? So maybe they are very efficient because in the end, their genes are very simplified. Okay? They are highly efficient. Okay? And they do not need such a complex regulation that a multicellular organism has. Okay? So, you can discuss about these things many times. This is just what we said. Genomes contain much more than genes. It contains many things. Regulatory RNA, regulatory DNA, and so on. Okay, any question about this chapter? Is there any questions about this first chapter?
How about amoeba? Does amoeba have more genes than human?
No. Amoeba doesn't have more genes than human. But as you saw, there is a lot of. Where is it? Amoeba, they have a very big genome. But the amount of gene of amoeba is not higher than that of human. But we don't always know, why they have such big genome. We don't know. We really don't know. Maybe it's true that these animals, maybe they have a lot of junk DNA. It's possible that there are many copies that are not so important. But maybe it's possible that since it's only one single cell, it is very sensitive to attacks by chemicals or whatever. So maybe it needs many copies of the same gene to be sure that always one functional copy is always functional. This is one possible reason. We don't need this because we are made of many cells. And then when we have dead cells, then we can remove them and we replenish them. But the amoeba, she cannot do that. It cannot do, right? So maybe this accumulating a lot of DNA, it's like maybe the equivalent is that it's almost like a ginkgo. Yeah, you keep a lot of money in your bank. You don't use it in your wallet, but you have a lot of money in your bank in case you run out of money. So maybe the amount of DNA for amoeba is the same. It's DNA that is there because if the DNA of amoeba gets damaged, it always has copies that can work. That's possible. But in the end, we don't know for sure these things. Okay? Any other question? By the way, this very strange name I told you, Saccharomyces cerevisiae, written here, right? Escherichia coli. This is the complete name of this animal. Hi.
Cool. Since there are a range of nucleotides per genome, per haploid, does that necessarily relate to size of like the animals had like a range of genomes per haploid, right?
Yeah.
Same with fish, and fish can be like your normal snapper, a whale, right?
Whale is not a fish.
Well, sorry, a whale's a mammal. I apologize. Okay,
It's okay.
Let's take it down. Okay, human and a whale.
Yeah.
Is that necessarily related to size of like number of nucleotides?
No.
No. Just
There is no clear relationship between the size of the genome and the size of the body. But that being said, for one organism, so for example, if you think the liver, your liver, for example, scientists have found that most of your cells have, as you said, 23 pairs of chromosomes. But some cells, especially in the liver, they have more than that. Instead of having pairs, it has maybe 4 pairs. So normally, how we wrote in biology is most of our cells are 2n. 2n means each chromosome has 2 pairs. Okay. But some cells in the liver can be 4n, or even 8n, or even 16n, 32n. And we don't really understand why some cells in the liver accumulate so many copies. And it might be because the liver is a highly metabolic tissue that is submitted to a lot of stress. So this could be a way by which maybe hepatocytes, which is the name you give to the liver cells, they can protect metabolically themselves by accumulating so many copies of the same gene. But what also was observed is that maybe the 2n hepatocyte may look like this one, and then with a normal nucleus. A 4n hepatocyte may be slightly larger with a nucleus about twice the size. And for 8n like this, it may be even bigger with a big nucleus. And then there are all sorts of in between. Like you have one hepatocyte like this, but actually it has 2 nuclei. But usually the size of these hepatocytes will be a relation of the amount of DNA it contains. But if you want to compare different organisms, it's not always like this. Like for example, amoeba is very small and it has a huge amount of DNA compared to human. Any other questions about this chapter? No? Don't hesitate. Right. So, the next chapter we are going to start. No. That's not the next chapter. Okay. And we are going to look a little bit at what our cells are made of. Not how they look like, but what substances they are made of. We are going to see the chemical bonds, small molecules in cells, and macro molecules in cells. Macro molecules means huge molecules. Okay. chemical bonds. Cells are made of relatively few types of atoms. You have all in your mind the Mendeleev table, the chemical table, right? But if you look at what elements living matter is made of, it's made of a very relatively small amount of these elements. Very basic, that's an atom. It has a nucleus and it has a cloud of electrons, okay? I think that this is known. So the nucleus of the atom is made of neutrons and protons. Protons are positively charged, and electrons, which are kind of gravitating around the nucleus, they are negatively charged. These are electrons. One hydrogen atom, as you probably already know, it's made of one proton and one electron, which means that one proton can be considered as a hydrogen atom who has lost its electrons. Okay, this is like a school chemistry. Now, I let you on with this one. You know what is a molar solution? You all know what is a mole, right? Yeah, okay. So, the outermost electron determines how atoms interact. So, this is what I said in the beginning. If you actually compare what the Earth is made of and what living things are made of, then you can see this is completely different. Mainly, the Earth is made of silicon. A lot. We are not made of silicon. But you can see that what we are made of is mainly hydrogen, carbon, oxygen, nitrogen, a little bit of calcium, sodium, and potassium. Be careful, in Japanese, potassium will be kalium, but in English it's potassium. Be careful, okay? And phosphorus. So it's completely different. But if you think about this, the structure of carbon is actually not so different from the structure of silicon. In terms of how many electrons it has in its outer shell. It's actually very similar. We will see later. So look at this one here. So if you remember from your very early chemical course, there are different electron shells. You know, the first one is closer to the nucleus, and then it goes outward. Okay. So hydrogen has only one electron in the first shell. Helium, which is a perfect gas, it has two. It's very stable. So its outer shell is completely occupied by the maximum number of electrons. Carbon, this is what I wanted to say. Carbon, the first shell is filled up, but the second shell has 4 electrons going around. And if you compare that with silicon, it's actually not here, but silicon also has in its out. It's not the same shell, but silicon also has 4 electrons in its outer shell. Okay? So you may kind of wonder what is the difference between silicon and carbon. Nitrogen has 5 electrons. Oxygen has this amount, 6 electrons. Neon is completely filled up. It's also a perfect gas. Okay? It's very stable because it has its complete set of electrons. Okay? So essentially, if the outer shell is not complete, or if it has just maybe one missing before being complete, it's usually very reactive. So for example, what oxygen, in the chemical reaction that oxygen do, what will oxygen will try to do? When oxygen reacts with other elements, what will oxygen want to do?
Oxygen deprive electron.
Yes, oxygen will want to take electrons from somebody to get 2 more. It wants to have 2 more electrons so that it's happy, it has its outer shell filled up, and he's a happy man or a happy lady, whatever sex of the oxygen atom, just to be equal and everything. What about magnesium? What will magnesium want to do? What will magnesium want to do? Or what will sodium want to do? Maybe sodium will want to collect 7 electrons. No, right? It wants to lose. It wants to lose its electron so that it has its outer shell, lower outer shell, filled up with the maximum amount. So this is usually the rule. So among these atoms here, which is the most commonly associated with sodium in the daily life, which other elements on this list is usually sodium thought of associating with? You are probably, especially in the gakushoku, every single day you are eating a lot of it, because in the gakushoku there is a lot of it.
Salt.
Salt. What is salt?
Natrium ion and chlorine ion.
Yes. Sodium ion.
Sodium.
Yes, I know. It's difficult. Sodium ion
Sodium.
and chlorine ion. So what it wants to do, sodium will just give to chlorine its electron, chloride will become Cl minus, sodium will become Na plus, and they will form a salt, and that's very easy stuff. Some atoms, they just don't want to give their electrons, they just want to share their electrons with somebody else, okay? So they form a covalent bond. It's by sharing the electrons they form a covalent bond. This is very easy. Sodium chloride, as we have seen before, in this case it's not a sharing of electron, like for example this O2, for example, so a molecule of oxygen, they are sharing their electron, but an ionic bond, like the one you have in table salt, sodium chloride, then it's a transfer of electron, okay? It's completely transferring. One become a negative ion, the other one will become a positive ion, and usually the attraction between the two resulting ion is very, very strong. Okay, and usually it forms crystals. Okay, so that's the stuff. And as I told you, you may remember that each column, actually the amount of electrons in the outer shell will be the same in the same column. So it means that the outer shell of carbon has 4 electrons, and it's the same as the outer shell of silicon. It also has 4 atoms. But that means it has 4. What does it mean? It has 4 electrons in its outer shell. How many partners can maximum carbon do with other elements? How many covalent bond can carbon do? Because all our molecules, they are based on carbon making covalent bond with other molecules, with other elements. How many covalent bond can carbon do? 4. How many covalent bond can silicon do? 4. Why are we not made of silicon? Because theoretically, you could make a protein based on silicon. It makes exactly the same number of bonds as carbon. All the nucleotides, all the amino acid, everything you could replace with silicon. Theoretically. So why not? Actually, there are some crazy people who think that there is a life, well, maybe it's not so crazy, that there is a life in space, it's maybe based on silicon. So you have a very stiff, sand-made Martian somewhere out there. You know, the truth is out there. So,
silicon covalent bond is weak.
silicon,
Covalent bond between silicon and hydrogen is weak. Yeah, silicon and silicon.
Yeah, it has to do not with the number, but how strong these links are. Actually, most of the molecules made of silicon, they are very hard, unyielding molecules. It makes rocks, it makes sand. So this is a bond that is actually not weaker, but pretty strong. And the molecules made of silicon, they have much less freedom to move than the one made of carbon. Okay? So it's not really how many bonds you can make with your partners, but how flexible these bonds and how modifiable these bonds can be. And carbon is actually much more reactive than silicon as well. Okay? So anyway, that's the thing. And mainly, if you remember the list earlier, we are mainly made of these things, but we also have quite a lot of these ions, especially sodium, magnesium, potassium, calcium. These are very important for the function of our cells. Hydrogen, of course. These are also present in our cells in small quantities. For example, iron. Mainly, where is your iron? Where is your iron in your body?
Red blood cells.
In red blood cells. Why a vampire is actually probably not feasible. Why a human being a vampire is not. Maybe not possible. Maybe it's possible. You have to think. Association of ideas. A vampire is something who eats blood, right? It's somebody who eats blood. So theoretically, if you eat blood, you can be a vampire. Can you eat blood? Can you drink the amount of blood in a human? And if you do so, will you be okay? I know it's a very strange question. You know these vampires, they eat blood, right? I am not mad, right? I am not mad. I assure you, I am not mad. So actually, if you really drink a lot of blood, like vampires are supposed to do, you are going to be very sick. Because actually, having a lot of iron, you will get iron poisoning. So it's actually very, very bad to have too much iron. Although iron is very important, why you have a lot of iron in your blood? Why is there a lot of iron in your blood? Where is the iron in your blood, first of all? In which cell contains the iron? You. どこにある鉄は血の中に赤血球ね。Why there is a lot of iron in your 赤血球? 赤血球 is red blood cells, of course. Why do you have a lot of iron in your... In which molecule? Well, one step by one step. Which molecule contains the iron?
Hemoglobin.
Hemoglobin. What is the function of hemoglobin?
Hemoglobin carry oxygen.
Yes, it carries oxygen. And for that, it actually needs to be associated with iron. Okay. Nice. Covalent bonds formed by the sharing of electrons. We know that. I'm going to sail through this chapter because you all know. Two hydrogen atoms, they form a molecule by sharing their electrons and form a covalent bond. Okay? And you have oxygen, it makes two covalent bonds with partners, nitrogen makes 3, carbon makes 4. And then you have a nice network of, atoms by actually combining all these elements together. But there are different types of covalent bonds. The one in ethane is one single covalent bond, which means that it can sort of pivot, But if you have a double bond, it's less yielding and the molecule will look like a flat sheet. Okay? This is actually quite important for the, for the remaining of the lecture. So, but some molecules are like this is oxygen, and it's made of 2 atoms of oxygen, so it's a molecule of oxygen. And what should I say? The distribution of electrons that are shared between the 2 atoms is equal, because each atom of oxygen will sort of equally share the electrons. But in water, because oxygen, if you remember, wants to have electrons, then it will kind of attract to itself more of it than hydrogen does. So actually the molecule here is actually polar. So there is a little negative charge here because the distribution of electrons is a little bit more around oxygen, and there is a partial positive charge in this region. And this is shown by delta, which is a partial negative or partial positive. Covalent bonds vary in strength. Ionic bond forms by the gain and loss of electrons, just like the one in table salt. We already know that. And if you can actually look at the atomic level, what happens in a crystal of salt, you actually see a very nice lattice. It's called the lattice in English, where you have a nice organization between each of the ions of the salt. Okay? That's usually not called a molecule, this is called a salt. So when you say salt, chemically be careful, because salt doesn't always mean what you put in your food. Salt can mean a lot of things. Okay? Like magnesium chloride is also salt. Ammonium hydroxide is also salt. Okay, so be careful. Non-covalent bonds help bring molecules together in cells. So not only you need covalent bond to make a molecule, so if you think one protein is one molecule made of many amino acids bound together by covalent bonds. But very often proteins, they associate with other proteins. And even though they don't always make covalent bond together, they can associate because together they make a lot of non-covalent bond together. That is because in the molecule there may be a lot of partial positive and partial negative charges on some of the atoms, which will make them ready to associate with other atoms of a different molecule. For example, you have, let's say, protein subunit A and protein subunit B, and they are good match for each other because they will fit like a lock and a key. You can see that these positive charge here will associate with this negative charge, positive, negative, and then it's a really good match. And even though one single non-covalent bond, maybe this one, is not very strong, if you have many of them, it will be very strong. Okay? It's just like this. Okay? One little link is not very strong, but when everything is tied together, you can pull until tomorrow and you are going to have pain in your arms. Okay? Hydrogen bonds are important non-covalent bonds for many biological molecules, so we are going to see what are these. I'm going very fast. If you want me to go smaller, you have to tell me, can you go smaller, please? Slower, please. So we are going to see what are these non-covalent bonds in organic molecules, and one of the most important one is the hydrogen bonds. A hydrogen bond forms because the hydrogen, notably in water, has a partial positive charge, and therefore it will kind of form a hydrogen bond with another atom of another molecule that has a partial negative charge. So the two attract each other. And one single hydrogen bond is very weak in terms of energy. But if you have a lot, then it's pretty solid. Okay? This is why when you spill water, it doesn't form single molecule of water. It forms a puddle of water. Okay? Because this is of the hydrogen bonds of water. So this is just the length and the strength of some chemical bonds. A covalent bond has a length of about 0.1 nanometer. A non-covalent ionic bond is about 0.25, and the hydrogen bond is 0.17. So it's quite tight for a non-covalent bond. It's quite tight. It's actually, the length is actually smaller than that of ionic bond. So it's quite small. And you can see the strength in water, because let's check in water because we are made of water. So the strength of the hydrogen bonds within our molecules will probably be very similar to this one. And then in kilocalorie per mole, it's very small, very not so strong. And if you have many of them, then it's quite strong. Okay. All right. Some polar molecules form acids and bases in water. So, for example, acetic acid, also vinegar, acetic acid. This is not water, but you can see the structure of this triplet of atoms is a little bit similar to water. So you have carbon, you have oxygen, and oxygen will sort of attract a little bit the electron of carbon, and will attract the electron of oxygen, of hydrogen. So there will be a delta minus on the oxygen, and there will be a delta plus on the hydrogen. So what happens is that actually this hydrogen will be tempted to go away. Bye. I'm giving you my electrons. I will go somewhere else and see if the grass is greener somewhere else. So then this hydrogen will go and may associate. It's reversible. Okay, it's reversible, and this hydrogen with this proton essentially will actually react with a nearby water molecule to form a hydronium ion, H3O plus hydronium ion, and then what results is the acetate ion, which essentially the oxygen is happy because it has completely taken the electron of hydrogen. So this is why it's an acid, because it's able to give protons into the solution. And this is actually water is an acid, and water is also a base, because water can actually reversibly at any time make both hydronium ion and behave as an acid, but it can also make hydroxyl ion and behave as a base. And in water you have all these things always happening. Okay? These things always happen. Alright. What's the time? Can I finish the chapter today? No. That's it. Finish for today. This is very quick, but this is easy, right? Everybody knows that. Sorry, わかった?ね、わかりやすいね。わかった。OK。ありがとうございました。